and welcome back to Neuropsychology. So in the last video, we talked about different kinds of spatial behavior, specifically route following, piloting, caching behavior, and dead reckoning, and we went over some testing that is used in the lab in animals. So in this video, we are going to dive a little bit deeper into a structure that is very important for spatial memory. So we're going to talk about the hippocampal size in taxi drivers versus bus drivers and how this is connected to spatial behavior. And we're going to talk about three special cells in the hippocampus, which are the place cells, the head direction cells, and the grid cells. Okay, so let's start off with the London taxi drivers. So this really cool study investigated the hippocampus in relation to topographic spatial memory. So what they did is they recruited licensed London taxi drivers and also bus drivers. And that these taxi drivers had to have an extensive spatial knowledge of London to be able to obtain this license. Whereas bus drivers always had to set a specific route. So they put all of these participants in the MRI scanner and they took detailed images of the brain and they looked specifically at the hippocampus. So what they found was that taxi drivers had increased gray matter, aka neurons, in the right and left hippocampus. However, bus drivers did not display hippocampal changes similar to those observed in taxi drivers. So the hippocampus is really important for the spatial navigation and topographic memory. So in this picture right here, you can see the hippocampus right here and also on this side. So this is a coronal slice. If you slice the brain right here, and you can see it right here. Okay, so let's talk a little bit more about this structure. So the hippocampus is a really important structure in the medial temporal lobe of the brain. So here you can see it from the side or the lateral view. And we have two of them, so we have them bilaterally. So the name hippocampus literally means seahorse in Greek. And as you can see, it looks exactly like a seahorse. So here you have a seahorse, and here you have a hippocampus that is cut out of a human brain. So these two bilateral hippocampi, they are part of the limbic system, as we learned in earlier videos. And the hippocampus has different subfields. So here you can see the different subfields. So we start off with the dentate gyrus, and after the dentate gyrus, we have the series of the corneomonas areas. So we start off with CA4, as you can see right here. Then you have CA3, CA2, and then you have CA1. So these CA areas are densely packed with pyramidal cells that are similar to those which you can find in the neocortex. And then after CA1, there are projections to the subiculum. So the subiculum is actually not a part of the hippocampus, but it is part of the whole hippocampal circuit or hippocampal formation. Okay, so studying spatial behavior in animals has a lot of advantages. So animals are really good at solving spatial tasks, and as they do, their single cell activity can be recorded with single unit cell recordings. So single cell recordings have led to discovering a remarkable variety of neurons that seem to be involved in spatial behavior. Some cells indicate ongoing locomotion, some signal boundaries such as walls, edges, or tables, and others keep time or track movement during either running or during rest periods. So in the next couple of slides, we are going to talk about these cells. So specifically, we are going to talk about place cells, head direction cells, and grid cells. Okay, so let's start off with place cells. Place cells are cells in the hippocampus that discharge when the animal enters a specific location or its environment. So within a short time of a rat being in a novel environment, hippocampal formation place cells will begin to discharge in relation to the animal's location. So if the rat is walking on a straight path, active place cells are more likely to code by direction as well as location. So if the lights are turned off after the animal has explored its new environment, place cells maintain their activity relative to the previously visualized cue locations. So if a rat is removed temporarily and then returned to the environment after these environmental cues have been changed, these place cells will modify their activity to represent the new environment. Okay, so let's look at this picture at the bottom right here. So you can see on the left here, you can see how this rat was moving through this specific space. So the lines are literally how the rat was moving. And then in the right two pictures, you can see, so in the middle picture, you see the initial firing of one specific place cell for this position in the room. So this square is the room, and whenever the rat was in this room, this specific place cell would fire. And then after doing that over and over again, you can see that it gets a little bit more accurate. So every time he's in that location, this specific cell will fire. Okay, so let's look at the picture on the left. So this is a very schematic picture of a maze. And here you have a little rat. So here you see an example of 8 different cells being recorded. So every color is one cell. So 1, 2, 3, 4, 5, 6, 7, 8. So for example, the purple or pink right here represents a place cell that fires only when the rat is in this location of the maze. And then, for example, the blue will represent another place cell that fires only when the rat is in this position of the maze. Okay, so moving a few visual cues does not change a lot in the firing of these place cells. However, if you rotate all the room cues, so for example, if you have a poster on every wall, if you rotate all those posters, then the cells will adapt and discharge with respect to the cue's new location. So it will technically think that it is in a new room. If a rat is exploring a maze for food, for example, some place cells will discharge when it is in a particular part of the maze. Moreover, these cells may discharge when, for example, the rat is intending to make a left turn, but not if this rat is intending to make a right turn. Place cells prefer visual cues that you could see in the environment. However, although these place cells prefer visual cues, they can also be influenced by olfactory, vestibular, tactile, and auditory cues. So, for example, a blind rat, the place cells of this blind rat will still respond to cues that the animal discovers by, for example, touch. So again, here you have a room, and here you have the rat right now. So the lines are everything where he went. And what this orange here tells us is that this is a recording for one place cell or a couple place cells nearby each other. And this place cell will fire every time the rat goes back into this room. So let's say the place cell is located right there. It's not firing when the rat's here, but then when it moves all the way back here, it will start firing again. So if an animal is placed in an environment with only one visual cue, then the single cue will determine where place cells will discharge. If that single cue is then removed, the cells will continue to discharge, but the location at which they discharge begins to drift. So if a visual cue that influences the firing of these place cells is moved unpredictably relative to other cues, then the place cell might stop responding to that specific cue. So place cell activity is closely linked to an animal's ability to move. So for example, if a rat is restrained or held in someone's hand, these cells will stop firing or discharging. So place cells can be recorded in structures other than the hippocampus, but only the hippocampal place cells appear to have this special versatility that allows them to change activity in response to changes in environmental cues. Okay, so moving on to the second one, which are head direction cells. So head direction cells are cells that indicate direction. Or specifically, direction of the head. So these cells discharge or are active or fire whenever a rat points its head in a particular direction. So different cells have different preferred directions. So for example, one head direction cell might discharge whenever the rat points his head to the west, and another cell or another head direction cell will discharge whenever the rat points his head towards the south. So the firing of the head direction cells is not related to the position of the animal's body or its trunk, but only to its head. So it also does not depend on whether the rat is moving or not. So head direction cells do not adapt as time passes, but they maintain their rate of discharge as long as the rat's head position is pointing in a preferred direction. So for example, let's look at this picture. So let's say we are recording just one cell, and this one cell will only fire when the head is turned that way, and it will fire a little bit less when he looks that way, and it will not fire when he looks the other way. However, if we look at another or a different head direction cell, it might be the complete opposite. So these head direction cells are similar to, let's say for example, a compass needle. So they fire as long as the head is facing in a direction. So a head direction cell is not activated by the presence of a particular object in the environment, but simply only the direction of the head. Surrounding cues, however, can influence head direction cells. If a rat is taken to a novel environment, its head direction cells will quickly develop a preferred orientation there. If the rat is then removed from that environment, and then if you rotate all the cues, and if you put the rat back, then the head direction cells will also rotate with those cues. So if the lights are turned off, head direction cells maintain their tuning for many more minutes. And when a rat is allowed to explore two environments connected by a tunnel, head direction cells will maintain the same preferred direction for both environments. Okay, so lastly, head direction cells are not limited to orienting the animal in a horizontal plane. They maintain their directional tuning when the animal climbs up and down as well. So these cells will also continue to discharge when the rat is restrained, unlike place cells like we just talked about. So whereas place cells may fire in one environment but not in another, head direction cells are locked into a network that is constantly active. And it literally only depends on the direction of the head or where the head is pointing at. Okay, and last but not least, the grid cells. So grid cells are a little bit more complicated to understand. So grid cells fire at regularly spaced nodes that appear to divide the environment into a grid. So each grid cell discharges at irregular intervals, and these discharges will technically form these nodes that are points of, let's say, like triangles. So the grid stays the same even when the animal changes direction, movement, or speed. So different cells at the same location have the same grid spacing and orientation relative to the environment. So let's look at this picture right here. So here we see the place cell that we just saw. So if the mouse is moving, like these lines are showing, a place cell will only fire when this rat is in a specific location. A grid cell is a little bit similar. However, this one cell will fire in this grid-like formation. So every time it comes here or there or there or there or there, it will fire. So a place cell tells us where an animal is in the location, and a grid cell will tell us kind of what the room looks like. So it forms a grid of the room. So again, grid cells are cells in the hippocampus that fire in many locations, and they form this like virtual grid. This firing pattern is invariant in the face of changes in the rat's direction or of his movement of speed. So in this image, the room would be round, and then this specific cell will fire whenever the rat is at any of these locations. Okay, so let's do a little recap. So as you can see in A and B, you see the place cells that fire in this specific spot in the room, irrespective of orientation. So this is a round room. Whenever the rat is in this location, the place cell will fire right there. In C, you see head direction cells. So head direction cells discharge when the rat heads points in a given direction, irrespective of its location. And in the last one, you see the grid cells, which is what we just talked about. Okay, so these cells are they're in more regions than just in the hippocampus. So the brain regions that house place cells, head direction cells, and grid cells are actually largely different. So in this image, you can see that the place cells are right here. So in this image, you can see that the place cells are most often recorded in the entorhinal cortex, and in subiculum, and also in the hippocampus. And head direction cells are often recorded in the lateral mammillary nuclei, or in the anterior thalamus, or cingulate cortex, parasubicular regions, and also in the hippocampus. And then the grid cells are mainly located in the entorhinal cortex and the hippocampus. So although the relations among the three systems are not 100% understood, investigators have established that the head direction system mediates navigation in relation to the animal's own location, and the place cell system mediates navigation by using environmental cues, and the grid cell system signals the size of a space and the animal's position within that space. Okay, so that was the end of this video, and I will see you guys back soon for the next one.